This video tutorial is about anaerobic respiration in eukaryotes. Respiration in the absence of oxygen. If oxygen is absent, oxygen cannot act as the final electron acceptor at the end of oxidative phosphorylation. Protons diffusing through channels associated with ATP synthase are not able to combine with the electrons and oxygen to form water. The concentration of protons increases in the matrix and reduces the proton gradient across the inner mitochondrial membrane. Oxidative phosphorylation ceases. Reduced NAD and reduced FAD are not able to unload their hydrogen ions and cannot be reoxidized. The Krebs cycle therefore stops, as does the link reaction. For the organism to survive these adverse conditions, glycolysis can take place, but the reduced NAD generated during the oxidation of triose phosphate to pyruvate has to be reoxidized so that glycolysis can continue. These reduced coenzyme molecules cannot be reoxidized at the electron transport chain, so another metabolic pathway must operate to reoxidize them. Eukaryotic cells have two metabolic pathways to reoxidize the reduced NAD. Fungi, such as yeast and plants, use the ethanol fermentation pathway. Mammals use the lactate fermentation pathway. Both of these pathways take place in the cytoplasm of cells. Figure 1 on the slide in front of you shows the fate of pyruvate under anaerobic conditions in yeast cells. This is known as alcoholic fermentation. Each molecule of pyruvate produced during glycolysis is decarboxylated and converted to ethanol. This stage in the pathway is catalyzed by pyruvate decarboxylase, which has a coenzyme, thiamine diphosphate, bound to it. The ethanol accepts hydrogen atoms from reduced NAD, becoming reduced to ethanol. The enzyme ethanol dehydrogenase catalyzes the reaction. In the process, the reduced NAD is reoxidized and made available to accept more hydrogen atoms from triose phosphates, thus allowing glycolysis to continue. We're now going to look at the lactate fermentation pathway. Lactate fermentation occurs in mammalian muscle tissue during vigorous activity, such as when running fast to escape a predator, when the demand for ATP for muscle contraction is high and there is an oxygen deficit. Pyruvate produced during glycolysis accepts hydrogen atoms from the reduced NAD, also made during glycolysis. The enzyme lactate dehydrogenase catalyzes the reaction. There are two outcomes. Pyruvate is reduced to lactate, and the reduced NAD becomes reoxidized. The reduced NAD can accept more hydrogen atoms from triose phosphate during glycolysis, and glycolysis can continue to produce ATP, or enough ATP, to sustain muscle contraction for this short period of time. The lactate produced in the muscle tissue is carried away from the muscles in the blood to the liver, where more oxygen is available. The lactate may be either converted to pyruvate, which may enter the Krebs cycle via the link reaction, or recycled to glucose and glycogen. If lactate were not removed from muscle tissues, the pH would be lower, and this would inhibit the action of many enzymes involved in glycolysis and muscle contraction. Neither ethanol fermentation nor lactate fermentation produces any ATP. However, because this allows glycolysis to continue, the net gain of 2 molecules of ATP per molecule of glucose is still obtained. Because the glucose is only partially broken down, many more molecules can undergo glycolysis per minute, and therefore the overall yield of ATP is quite large. However, for each molecule of ATP, the yield of ATP via anaerobic respiration is about 1/15 of that produced during aerobic respiration.